When I was interviewing for postdoc positions, it became clear to me immediately that the Green Center for Systems Biology would offer me an incredible environment to work with a team of interdisciplinary experts. I must say, I could not have chosen a better place for my postdoctoral training.
The Green Center aims to develop approaches to dissect biological complexity, leading to the alteration and even the engineering of biological systems at the cellular and the molecular scales, ultimately leading to fundamental understanding as well as the ability to target human diseases. Drawing inspiration from the engineering fields, the vision from the Green Center is to be able to modularly compose biological systems into circuits that we can understand, predict quantitatively, and ultimately program. To be able to do this requires collaborations and new ideas across multiple disciplines of science and engineering, both theoretical, computational, and experimental. The center is bringing our framework to bear on two important biomedical problems: infectious disease and cancer metastasis. Both challenges serve as excellent model systems of biological complexity for us to apply our circuit framework to.
I study how cell shape regulates the molecular circuitry of oncogenic RAS, which is a very notorious human oncogene. My hypothesis is that the oncogenic effect of RAS is enabled by very thin cell membrane protrusions called ruffles and lamellipodia.
I'm Scott Saunders. I'm a Distinguished Fellow in the Green Center for Systems Biology. This unique position is allowing me to build my own research group immediately after my PhD. My research focuses on how bacteria work at the molecular level. This is a huge interdisciplinary challenge involving computational design, systems-level thinking, and organism-specific knowledge. I'm very excited to be at UT Southwestern, where I can collaborate with colleagues from the Green Center for Systems Biology, Department of Microbiology, and clinicians who directly work with resistant bacteria from sick patients.
As someone working towards becoming a physician scientist, I have a really fortunate opportunity in working at a center that has such a really strong basic science program while also being a medical center.
I've loved training here in the Green Center. Our team is so broad in their scientific interests.
I'm broadly interested in understanding the relationship between genotype and phenotype. My lab works on this problem in bacteria.
My lab works on creating a computer programming language that will allow us to describe the circuitry of organisms at the levels of molecules, cells, and tissues.
In my lab, we approach the problem of biological complexity from the perspective of non-equilibrium statistical physics.
I've been developing a light-inducible CRISPR-I-based system that I can use to introduce transient repression of specific bacterial genes in continuous cultures in the evolver.
My dissertation work focuses on how metabolic genes work together to promote growth in E. coli. So using biological tools like CRISPR interference, I can rewire these metabolic circuits and quantify how my new engineered system behaves.
The computational biology graduate program here at UT Southwestern allows our students to train in a rigorous curriculum in mathematics and computer science at a first-class academic medical research center. Our students receive training in mathematics, computer science, machine learning, information theory, and statistics. And this coursework is bolstered by a close relationship with the BioHPC. The BioHPC is our first-class parallel computing center, and it's staffed by really great technical staff that helps support our students and also offers coursework in computational tools and tricks.
In my research, I'm using this framework to study high-order epistatic interactions between the resistance-conferring mutation in TEM1 beta-lactamase.
I feel I've learned as much about communication as I have about microbiology and computation. These skills taken together will absolutely shape my future in science.
We have a uniquely collegial and collaborative environment, and I really like that the Green Center is kind of unified around this vision of understanding the architecture of cellular systems and demonstrating that understanding by going and making quantitative predictions and designing new cellular systems in the laboratory.
Interdisciplinary research collaboration is quite enabling. I would say it is the essence of modern science.
Our approach offers interdisciplinary tools, ideas, and applications to make the breakthroughs necessary for progress in biomedical science.